the impact has been extraordinary of CARs. And just looking at it currently, there are more than 300 ongoing trials of such therapy, mostly in the United States, North America, mostly United States, some in Canada. There are enormous number of trials going on in China, and in Europe, less than 10% of all CAR trials actually take place in Europe, although there are some important leaders in the field, especially in the allogeneic CARs. We went through the various types of CARs. The one which made history is what we call the autologous CARs, which refers to using the patient's own T cells. And of course, the two drugs that have received two CARs FDA approval, they've been when the patient's own T cells have been manipulated and together with a particular target. In both cases, these were targets that are known as CD19, one on acute lymphoblastic leukemia, acute lymphoblastic leukemia in pediatric age group, which means from 3 to 25 years, and the others for adults with diffuse large B-cell lymphoma who are not going or not eligible or have decided for some reason not to undergo a transplant. So here we're using the same target. But that is where the initial exciting work has come from, but this has opened up the entire field and multiple other targets for virtually every other disease. It's enormously complex, and the science now is finding the correct target. There are problems. You have to be sure when we're trying to stimulate the T cells that we don't give a target that is antagonistic to T cells, so we have to find the right targets, but virtually this has opened up the treatments of all lymphoproliferative disorders, which are the leukemias and acute and chronic leukemias, ALL, CLL, the lymphomas. In the lymphomas, we also put myelomas. Myelomas are basically a lymphoproliferative disease, just the more mature cells, plasma cells. And trials are beginning in acute leukemias, in other acute leukemias, AML. The reason why the lymphoproliferative disorders have been such a starting model, in fact, why ALL has been the very first drug in which it was started on, really a rare disease in adults, and the trials were done there. Why? Is because we know that if you wipe out the target as a CD19 and you wipe out CD19 in a patient, so a patient doesn't have his B cells, patients can survive very well without it. That's the barrier, for example, looking at AML. If you do a target against AML, say a common target would be CD33, you can't give somebody CD33 and wipe him out because the patient will have no normal neutrophils to survive. That's been a barrier. So ALL was in fact an ideal target. We could target a particular antigen which also exists on normal cells, but we've learned from experience with using rituximab that patients can survive well without certain types of B cells for a period of time, obviously. So that's where everything started. And now the science and the art is to find targets. This needs a lot of manipulation. There's a lot of there's enormous work that has gone into it. Now, these are expensive medications, but then they cost nothing compared to the investment that has gone into it. To make a CAR therapy, besides the technological equipment, you need enormous teams at work. There's usually 3 or 4 major teams working on basic immunology, working on T-cell biology, the transplanters together, and the work that has gone into it. Financially, globally, it's only going to make sense if you do it, if this is going to become so popular and common that it will be done for many patients. Obviously, the greatest need and greatest potential, but at the same time the greatest challenge, is going to be in solid tumors, just because they're much more common. It's harder to find a specific target. And so the design now studies is, because they're multi-targeted, is to use several targets together in order to be sure that we hit them.